Okay, thank you. I'm going to preface these comments by pointing out that I certainly understand the need to move in this direction. I'm also on the Science Advisory Board of EPA, so the problem of how to move risk assessment forward is obviously an acute issue for them. Couple things that I recognize also, we've had very limited time this morning. We certainly haven't heard about everything, and there have been a lot of other meetings, both internally and of groups, to talk about how to move this process forward. But I think there are a couple of omissions that we haven't heard talked about today, and I want to talk about one of them in particular. First of all, what do we mean by what constitutes toxicity? What is an adverse effect? And we haven't heard very much about that. We keep talking about outcomes, and whether those outcomes are toxicity or adverse effects, there hasn't been a lot of discussion of. But from my own perspective, I guess it's the 800-pound gorilla, maybe in today's world it's the 1,000-pound gorilla in the room, is validation. And I'd like to talk about the validation. How do we validate these assays? It's come up in some of the discussions, but let's talk about a couple examples or case studies, if you will, that are of importance to me. So I work on the nervous system and I work on behavior. And if you think the slide showing the person putting wires on a panel was complicated, multiply that about 15 times, and then what you're looking at is the system of systems, which is the nervous system, which is multiple systems that interact and talk with each other, and how we're going to get some of these simple assays to validate the outcome of that, which is behavior. So how do we do that? If you just think about, we tried to do some of this a long time ago, interested in the problem of learning during learning deficits, memory deficits that occur with age. And when you start trying to do some of these kind of experiments, just thinking about things like gene changes, first of all, you have to define what's learning. Well, it's a dynamic process. The nervous system is very dynamic, and learning occurs in stages. And if you're just working with an animal or a human, by the time you're convinced that there's been learning, is at the time to look at the gene changes, the effect has already occurred. And so we're already sort of stopped at where do we stop and take these data points in order to see what kind of gene or protein or other changes there might be. If we're going to look at toxic endpoints that are morphological or pathological, the nervous system doesn't need those in order to have pretty serious behavioral deficits. We can be looking at things as simple as imbalances in neurotransmitter function, too much of one versus too little of another at the same time. So we would already be underestimating risks by using this kind of an approach. I think the other thing about that is that you're going to have different kinds of learning. Those are going to have different kinds of behavioral mechanisms. So again, I'm going to come back and ask, what is it we're actually trying to model here, and how do we go about extrapolating from very simple assays to something as complex as behavioral function? I think one of the things that strikes me as having listened through the morning is that maybe what needs to happen in these kind of programs is prioritize those endpoints or outcomes where you think validation is possible, and put more focus on those before trying to get into things like behavioral toxicity. The other point where the validation comes in from my point of view is with respect to cumulative toxicity. So it's kind of striking to sit and listen to increasingly reductionistic mechanisms, models of trying to predict toxicity, when the biology world is in a way going in the other direction. That is, recognizing more and more that most of the diseases that we're concerned about are really complex disorders. They're the interaction of multiple risk factors, some of which may be environmental, but there are a whole lot of others. And so maybe it's not just a toxic outcome that has to happen, but some other insult has to occur at the same time. And so how are we going to come to model these with respect to human disease? And really, isn't human disease what ultimately diseases and disorders, ultimately what we're trying to protect here in terms of public health? I think you have to couple that with a couple of other things. One, we spent a little bit of time this morning hearing about dose, the importance of dose. What I didn't hear any discussion of was the nonlinearity of dose effect curves. That doesn't just include toxicants. There are a lot of toxicants that have non-linear dose effect functions, especially at lower levels of exposure. How are we going to deal with those? You have to couple that, in the nervous system at least, with the nonlinearity of control of many systems. For example, in many neurotransmitter systems, there are actually U-shaped or inverse U-shaped curves that control behavioral function. So turning something up or turning something down, unless you know the context, can be very difficult to interpret. Couple that with similar effects with stress hormones like glucocorticoids, which have a huge impact on the nervous system, also operate by nonlinear kinds of dose effect curves. So your underlying biology is nonlinear, and many of the toxicants that we're interested in, certainly for the nervous system, also have nonlinear dose effect curves. The interaction with non-chemical stressors, I mentioned that before. And I think finally, coming back to a point that I made before, really I've heard a lot about liver tumors. I think moving these assays and worrying about cumulative risk has to be in the context of diseases and disorders, and that's really how we get to those, how you put together these different stressors that's ultimately of interest, and I haven't heard very much discussion about those. So for my quick 5 minutes, those are some of the things I try to see how we're going to make the leap. I think some of the efforts that we heard trying to understand relations between genomic changes, proteomic changes, metabolic changes, and ultimately function. It's ironic, this story about coming back finally to physiology. Our toxicology training program has people that come back from industry and say, when are you going to teach these students physiology? Because it was, in fact, taken out of the curriculum several years ago. And I know a lot of students who literally can't find their way outside the cell and don't understand how basic physiology works. Finally, with respect to the nervous system, there's a huge interest right now in how peripheral systems communicate with the brain in terms of things like immune challenges, infection. They can basically activate similar cascades in the brain itself. So there's a lot more brain, PNS, brain autonomic nervous system communication that goes on that's going to be very difficult to model and figure out. In the end, I have to ask whether or not some of these approaches, first of all, need some prioritization in terms of what's really achievable, but secondly, how we're going to get to whether we're going to be ultimately underestimating risk, because we may see things that in some of these assays don't show up as toxic, however we define it, but yet in the real world, when we combine them with the other risk factors for human diseases and disorders, they may indeed, at much lower levels of exposure than we've even thought, ultimately prove to be problematic. So I think those are some of the challenges.
Thanks, Debbie. Abby?
So I'll try to stay with things that Debbie has not mentioned. I think the things she has said are very important. But I also am a neurotoxicologist, and I do some of my work relates to developmental neurotox. And we are dealing with an assay right now that's approaching $1 million, and we have like 650 pups at least. Usually we have to have like 25 to 35 liters per dose level. So I'm kind of excited about the potential that computational toxicology might afford us, especially with respect to looking at molecular endpoints that are critical to specific pathways that are important to development and conserved across species, so that hopefully they might be more predictive of what's going on in humans, and allow us to get past just the apical tests that are also important, the functional endpoints, but to bring maybe some more basic biology to that. So I'm excited about the possibilities, and I've been asked to maybe focus on what I think are limitations, since we've heard a lot of the strengths. And when I think about strengths and limitations of this overall field and what we've heard about, I have to ask myself, well, it really depends for what purpose. So I kind of categorize these into 3 areas. One is developing hypotheses for further testing, increasing your probability of finding something that might be relevant, that you recognize needs to be tested further, that you can't stop with just the computational toxicology, the results, but you have to take that information and then test it in a biological system. The second one is to actually screen chemicals for further testing, or to determine if more focused testing should be added to a shorter term study. So you might find something that says that maybe in a 90-day study that you might be doing, you add several other endpoints because of what you might find in these screens. And the third is actually replacing in vivo testing completely for regulatory decision making. And for the latter two regulatory purposes, I'm not comfortable where we are right now. And I believe that one of the things that we really have to, that there's some things that we need to start populating into our database now in order for us to have any possibility of success for the latter two more regulatory purposes. And the concept of dose is really important, and that's not always entered into the database from what I can understand. And if any of that could be linked with some possible relationship to exposure, and I was speaking to someone who's on the panel who does a lot of exposure, that maybe you can speak about, is that something, you were telling me that we have occupational data on exposure on a wide variety of chemicals. Can we, if we're going to prioritize chemicals, can we somehow relate exposure in the petri dish with something that bounds us in terms of exposure? Because prioritization has to have dose related concepts in there for us to decide which ones to chase. You can always put a high dose in something and find a lot of effects, but I'm not sure whether chasing all of those is going to make a difference for public protection. Or in the absence of exposure data, can we somehow anchor those effects in the database against some metric of cytotoxicity, recognizing that we have to be cautious about cytotoxicity, because you could have, for example, a specific effect on the nervous system, like neuronal proliferation, which will cause cytotoxicity. But you could also compare that against other cell types to get a sense of whether you're having something that's specific. So I don't hear those sort of things of anchoring what we see in the petri dish that's being entered into this database against things that will help us to understand dose. I think the second thing that really has to happen is some criteria for data needs to be entered. So George was talking about the Miami criteria, and if I look at the publications, we're talking about starting, that was like 2001, and then it was adopted in like 2005. That's fairly recent. There's a lot of literature, and I would say that the toxicology literature is always behind the standards of the genomic literature in terms of the quality of peer review that happens. So, some kind of standard has to be in there before we put it all in, or else we're going to have a lot of junk data, and that's not going to help us move forward. Is there a way to weight the quality of that molecular data? For example, the NAS report on the 2007 NAS report on toxicogenomic emphasized that in a toxicogenomic experiment in which thousands of genes, this is a quote, proteins or metabolites are examined in a single assay, biological validation is important because there is significant likelihood that some changes in genes, proteins, or metabolites are associated with a particular outcome simply by chance. So upregulation of gene transcript detected with a microarray may suggest or hypothesize that something's happening, but you have to confirm that. And there's a range of data that I know of in the toxicology literature where sometimes you have papers where all they do is lots of microarrays and no confirmation. And even now, we're faced with regulators asking us, well, what about this data? It's it's used in the, public newspapers to prove that this means we've got an effect on genes that are related with autism. And we have to be very careful about making sure that this data is grounded in and strong. Debbie talked about validation with a training set of positive and negative controls. And I would say that the negative controls really have to be thought through, because you can't just have a negative control that has absolutely no activity, but something that would really challenge the system, so that maybe it's a liver toxin, and you want to see whether it's going to also affect something that's neurologic. I mean, how can we try to make sure what we're seeing is not just general? So finally, methods to address, well, metabolism is essential. Transparency and eventually standardization of culture conditions are needed, because what you might measure can really be dependent on the culture conditions. So, Debbie talked about adverse effects. What is a precursor event that might be adaptable, and which ones lead to adverse effects? So I say that computational toxicology is only as good as the quality of the data entered and the assumptions we make. And that's got to be based in biology, not purely statistics. And, I started my comments with talking about how excited I am about actually moving the field forward so that we can look at highly conserved molecular pathways. But then I worry that maybe we set up this whole assumption based on a flawed assumption, because does that mean that maybe we're ignoring the very things that are very important for humans, that make us humans? I mean, like, I think about the developing nervous system. A zebrafish doesn't have much of a forebrain. Are we missing out on things that are really critical to humans because we're so focused on only those things that are highly conserved in lower species? So I end on, what I did was I'm very excited, as I said, about these highly conserved molecular pathways. And I don't know, some of you geneticists in the room are, must know better than I do Eddie Rubin's group at Berkeley National Lab. And he did some stuff that had to do with the forebrain and some of the genetics. So I was interested in that. And what he did was he used computational approaches to find extreme evolutionary sequence conservation. And meaning that sequences that are the same all the way from human all the way down to fish. So he used computational techniques to do this. And then he used those to ask the question, this is, what is biologically meaningful? And he got only a 45% hit. Wow, that could be really good. That's really good, right? A 45% hit's really good if you can focus your research on those things that then allow you to ask and test something that's actually biologically meaningful. But if you're going to think about the second two criteria where we have to make regulatory decisions, then 45% hit is like flipping the coin. And we have to do better than that. So I think we're at the stage where we're at the first stage where we can use this data, computational toxicology, to really guide our hypothesis, develop hypothesis, and increase our chances. But we've got a long way to go, and some of the things that I mentioned are going to be absolutely necessary for us to move towards regulatory risk assessment decisions. Thanks.
Thank you, Abby. Ben, you had slides. Do you want to come up here and do that? Okay. Oh, and incidentally, in California, they don't flip a coin. It's an IOU.
Oh.
Well, I've really enjoyed the day, and I appreciate the opportunity to talk about some of the themes that I've heard emerge. And hopefully I'll be able to reiterate some of the things I've heard today, as well as also agree with some of the concerns that were already raised. I'm sorry for those of you that are sitting up front that can't see the slides, but I'll just read through them. I think we've seen a lot of new tool development, but I also feel very strongly that we need to leverage the tools that are already out there working. And to do that, we need to make the data more accessible to more people. I think a theme I've heard over and over today is that the modelers need more data, and the more data they have in terms of dose response and time, they can definitely develop better models. So I believe this field would be enhanced if we could get those data out there in a usable form so that people can mine them. I think we need to think about how to connect multiple data sets in what I call biological space. Each genomic tool that people are using is appropriate for one slice through biological space. But the best studies that I've seen on this field bring two different approaches together. And at that interface, I think it's very strong and powerful. I think there has to be a united commitment to the best tools and better user interfaces. I think there should be benchmarks, and I think even competitions. What tool works best for a particular problem? A tool that I've been able to use and was developed by a number of people, Trey Ideker uses it extensively, is it's a wonderful platform for allowing you to make sense of large amounts of data. These kind of bioinformatic tools allow you to visualize data in a way that tables just don't give you that information. I think we heard about some wonderful data sets, but I would like to encourage people to use the data that's already out there. The yeast Saccharomyces genome database is highly curated, and there's some outstanding responses to toxicants in that database. And since it's already curated, I think it would be outstanding if there was money available to incorporate that into Carolyn Mattingly's wonderful database that's grown beautifully. Right now, yeast is not involved. We also heard about C. elegans and zebrafish. I think that the use of zebrafish allows certain information that you couldn't get any other way. But what I think is wonderful, and if you haven't ever gone to these databases, is the amount of information at your fingertips. And if there would be a way to harvest that information within a toxicological setting, it would be a win-win situation. I was really impressed what I heard about the actor database and a wonderful approaches that are being used. But again, if we can leverage these other databases to bring information to that. One wonderful database is called the string database, which is a collection of protein-protein and protein-gene interactions. It's a highly curated data set in a bunch of different organisms, and it allows you to understand potential interaction partners by two hybrid, by tap affinity columns, by genetic interactions, even by literature. And so this is a wonderful database that's been put out there that, again, I think we could use to leverage other databases. I'll have to, again, back away from high throughput and maybe talk about medium or high density data sets, but I think there are some wonderful organisms that can be used, but they have to be used carefully. I mean, it's a yeast, you wouldn't want to do neurological testing in yeast, but if you had compounds that you believed were important for cell cycle inhibition and those kind of things, I think yeast would be perfect for that. Similarly, the ability to knock out every gene in yeast that's not viable, that will not cause cell death, combined with toxicant is super powerful. And the same holds for siRNA and C. elegans. You can knock down every gene in C. elegans by feeding them bacteria. If you can then screen the effects of those gene differences with toxicants, you have an amazing data set that you could link. Similarly with morpholino knockdowns in zebrafish, the use of inbred mouse lines or knockouts. And then there's some beautiful work that's coming out using lymphoblastoid cell lines from genetically diverse people to look at sensitivity to different agents. And so I think this idea that we didn't hear a lot about today, we're not all going to react similarly to the same toxicant, and I think we need to understand the genetic susceptibility. One big problem, I think, in taking this wonderful computational biology to the next level in risk assessment is understanding both the effect and the pathology in humans. And you can't get access to some of the tissue that you're interested in. You can't get a brain biopsy or a liver biopsy very easily. And there are three, and I just chose these out of the literature, three wonderful papers that suggest that peripheral blood can actually allow you to see not only exposure, but also an effect and a potential pathological effect. The first one was a wonderful piece of work by NIEHS where they looked at acetaminophen toxicity both in the rat and human, and they saw that in blood there was actually a signature of both exposure and also toxicity. This was a beautiful study out of Leona Sampson's group looking at infants born to women who are exposed to arsenic, and they actually could look at cord blood and could actually see a dose relationship with exposure to the mother with responses in the blood of the babies. And this has sort of been recapitulated in a more recent paper looking at people exposed to arsenic, again, using their lymphocytes. So I think this is a really a wonderful opportunity. I'm going to end with this last slide. We've talked and we've heard a little bit about it this afternoon, is we need to develop a priority list. And I didn't see any computational tools being used to attack this problem. And if you think about it, you could start from first principles. What are all the toxicants that we're exposed to? We could actually measure those and know. A lot of those toxicants are not that nasty, so we should weight those. Clearly, we're all exposed to drugs. For example, acetaminophen actually gives a lot of liver toxicity because of this, I think, genetic susceptibility. So I think we have to begin to weigh those things. Then clearly, we're most interested in human health and disease. And so the strongest linkages, and that's why I was really impressed with Carolyn's idea that there are interesting information coming from potential chemicals and disease. And if those could be weighted and made stronger, then we could put more emphasis on that. So I really would love to see the computational field begin to think about how to weight these priorities and attack that problem.
Thanks, Ben, and I wanted to thank all three of you for being really thoughtful and really provocative in your comments. There's a lot of stuff here in addition to our fairly generic questions for this session. Let me start by asking the folks who spoke today whether they have any reactions to what they've just heard from our three additional panelists, or other thoughts that they've had around integrating some of the things that you've presented with some of the things other people have presented to address some of these questions, or fatal flaws that you've suddenly seen, or new opportunities that have come up today. So I just want to give you guys the first opportunity. Richard.
So I want to just address several of the points very briefly. So the question I heard was, there was a worry about reductionism, but I would turn that around and say reductionism works sometimes, and sometimes it obviously doesn't. Where it does work is when we know a mechanism of action that can lead to some endpoint. Okay, and there aren't a huge number of those, but certainly where we know those, I think this is a true statement. Most chemicals, if you look at the big matrix of all chemicals by all pathways that one could measure in an in vitro system, that's going to be a very sparse matrix. So if we take the 10,000 chemicals or the 100,000 chemicals we worry about across all of those known pathways that you don't want to hit, that's going to be a sparse matrix. So we can essentially clear most chemicals of acting through those. And that lets us set priorities. Only the very small fraction that hit, we have to worry about. And maybe we go do the standard animal experiment at that point. Okay, but it certainly lets you take a lot of chemicals by a lot of things to worry about and bring it way down. Once you've gone beyond that, you probably have to start building models, and we've heard about some of these models that are have nonlinear interactions and feedback and all of that. Do you want to address it?
I'd like a clarification on all chemicals and all pathways. It would be a sparse matrix. And I'll pick up on something Steve Wiley said. There's no average cell. So if you take this cell, this cell, this cell, this cell, they're going to have different levels of different pathways. And I know from work, and actually I have colleagues, and I'm a co-author on a paper using some of the measuring a lot of the kinase nodes and looking at knock one out kind of models. And there are really vast differences between a liver cell and a mesenchymal stem cell, for example. So could you clarify what you mean by it would be a sparse matrix?
Okay, yeah. And it actually goes back to dose response, okay, which is clearly a very important thing. At doses that are, this is a great leap, at doses which are physiologically important, that are coming from accidental exposures to environmental toxicants, in most cell types, for most pathways, for most chemicals, that'll be a sparse matrix. That's my prediction. Okay, I don't have any data to back that up.
And it's linear, or I mean, because we heard about biphasic curves, and I've heard these things about toxicology. I'm not really up on them, but
If we're assuming that again, this is a very reductionist, that for most chemicals to cause most kinds of problems they cause, they ultimately have to interact with some target, an enzyme.
yeah, but if they kill some cells, but then
okay,
they,
but they're not killing cells by magic. They're killing ultimately by interacting with something. Okay, this is a
right.
faith-based
No, I'm
statement.
really just asking, is it, because an immune cell might respond one way,
Yeah,
and
no,
a
absolutely. And in
mesenchymal stem
fact,
cell might respond a different way, and the interaction of them may give you a completely
right.
unexpected
We've tested, I don't know, 20, 30 different cell types, human, primaries, whatnot, and you certainly see very different response, but it tends to be correlated. Chemicals don't do completely random things, cell type to cell type, at least at low enough doses that I'm going to worry about. Okay, so that it's a prediction. We can come back a few years later and
Just respond to
yeah,
that.
please.
I mean, in the brain, we have, for example, multiple dopamine systems. They control different behaviors. They have completely different kinetics, different control mechanisms. They're all dopamine systems. So that argument just doesn't even hold for the same neurotransmitter system in the brain. They operate very differently. So
No, but okay, all right, but again, it's not going to be some magical thing that happens to cause dopamine A versus dopamine
Tell
B.
that to people that have Parkinson's disease. They have the death of one of those nigrostriatal dopamine system. It's right next door to the one that controls executive function, which is completely spared. So
Okay, what we've done is we've tested against a bunch of dopamine receptors, dopamine, serotonin, and so on, and most chemicals don't interact with those. I mean, if this is one argument for another argument for this point, and then I'll drop this, is that if so many environmental chemicals on a daily basis, I've gone and actually counted through my house. I encounter a couple of thousand chemicals in my house every day. All right. If there was a high probability that matrix was not sparse, if all of those were hitting some pathway in my body, we would all be really sick. Okay. So it must be true that most chemicals at the sort of exposures we're getting are not doing too many bad things. Okay, all right. So in the average person, right.
Yvonne,
Okay.
you look like you want to weigh in on this.
I think I just wanted to add that like Rick said, everyone's right in this discussion. I think for the purposes of this workshop today, though, we have to realize that we're talking at the probably very high level at the assays and approaches that are not going to be either one size fits all or be able to address every possible toxicological question in every possible genetic background or disease background that an individual may have. And we have to realize that as toxicologists and regulation, we do jump over a lot of different important questions or step over, and I think that if we worry too much about whether high throughput screening or computational approaches or modeling will not be able to replace or do everything for everyone, will never be there. So I think what would be good for us to today to focus on what the tools and the approaches and the data that we have now can help us today and maybe tomorrow, not in the bright future. So I think some of these discussions are very good, and we should probably continue having them. But having the people who do the screening and having the people who have to apply them or think about whether that's a valid approach, I think we have to somehow be more at the ground level than try to make sure that every dopamine receptor in every cell may be a target or not.
Can I respond to that? Your premise is I guess I disagree with it. And this is why I brought up prioritization before. You could focus simply because there are finite resources. So it may be that this approach works great for some kinds of toxicity, and fine, you can focus down on some of those, but maybe some of those other resources or that pool of resources should then be devoted to other things to address other aspects of toxicity.
I agree 110%.
Okay, all right. So let me take the dopamine instance. Okay, so we've no, I'm sorry. Okay, but just as a random, for instance, okay, we certainly see chemicals, pesticides that many of us are exposed to. A few of them are hitting dopamine receptors at relatively low doses. I mean, I think we would all agree that if you worried about neurotoxicity or neural problems, those would probably be the ones you'd go after first, right? Could we at least agree on that? Be, but so how do you set priorities if not by using data? Okay.
From my point of view, that's part of the complexity when you try to approach the nervous system with some of these kinds of questions. I mean, in some cases, it may not be at a receptor level per se that you're going to see the effect, because the system will change its receptor sensitivity or number to address a problem, and it's just a slight imbalance. You'll never pick it up in an assay that's just looking at a receptor change.
Well, no, I'm looking at interaction with the receptor.
That's what I'm saying, but you wouldn't pick up some of these kinds of deficits in an assay like that. So A, I couldn't single out one neurotransmitter, and B, just changes at a receptor level per se are not necessarily indicative of what can go on in a system. You can have profound changes before you change anything at the receptor level that affect behavior, before there's even change in numbers or affinity at receptor level. So those aren't the most sensitive assays for sure.
Okay, let me make one more, jump in and answer and address one more thing, and then I'll be quiet. So that the issue of exposure, which is clearly important, I think, if we can certainly set priorities, I believe, based on the likelihood of exposure, not industrial workers, which is a whole different issue, but sort of average exposure. It's hard. We have a nascent program at the EPA, is the CompTox, called ExpoCast, which is kind of aligned with ToxCast, trying to come up with models where we can take the 10,000 chemicals or the 100,000 chemicals and rank those as what's the probability that you will be exposed to them at some level. Then coupled with that, we're also doing a combined modeling and experimental protocol where you're essentially saying, if we know that you have an IC50 at some target organ of 1 micromolar, okay, what would the dose you would have to take orally to get to 1 micromolar? And it's simply taking into account plasma protein binding and metabolic clearance and a few other things. Okay, so then you can go back and, for instance, go to NHANES. This is a little bit of a stretch, for instance, and say, okay, here's all the chemicals that are tested in blood, and we know what the average levels in the American population of those chemicals. Okay, what targets, how many of those chemicals are at the 1 micromolar level in, does the exposure level with the activity level sort of match? And if it turns out the average exposure that you actually see by doing biomonitoring is way lower than you would actually trigger any sort of molecular activity, then you probably don't worry about that. Whereas if the blood levels are significantly higher, then you do start worrying. So it's a priority setting approach based on some observed exposure levels.
Can I just respond to that? Because data from actually EPA itself, I think, is very pertinent to that. You run the risk of not taking into consideration cumulative risk from doses that are below threshold, which can act together even at below threshold doses, and some to become effective doses. And so how would you take that into consideration with that kind of a model?
I guess the answer is you walk before you run.
I guess I would say, isn't this the beauty of this system. I mean, basically, you know, here we've taken advantage of 50 years or so of reductionist approach to biology and toxicology, and we're summing them in a way that can only be done by the computer. I mean, that's really what the whole ToxCast thing is. So if you have these multiple exposures, now you know which ones ought to be summed or which ones ought to be considered together.
I thought he was saying that he was eliminating them from consideration if they didn't reach a certain level. Therefore, they were not
You down
to be considered.
prioritize.
Right. But that again, I don't know what down prioritize means because I don't know I don't know where you go as you go up the ladder. But certainly I don't think this is my statement before about underestimating risks by taking those kind of strategies and assuming that something's not problematic if it doesn't reach some exposure level associated with target organ damage. Doesn't mean it's not having an effect, especially in conjunction with other things that have the same common adverse physiological effect.
And I would say that this has been the first opportunity that anybody in a regulatory context has had to address that very question. You know, otherwise, well, I mean, in a systematic way, it certainly is. Yvonne.
Yeah, I'd like to turn to another question that we already had both brought up in the program and also by our panelists, which is of when do we know that the assay is good enough and whether it's helping us to interpret something, and maybe pose this question to our speakers in this afternoon, because we're collecting a lot of data and there are straightforward brute force approaches to just correlate everything with everything that we have data on in vivo, or try to think more about what does the chemistry bring and what does biology bring, or actually try to then put this into the context of physiology and molecular networks. But as we go up in scale of complexity, the amount of data and our ability to grasp all of this diminishes quickly. So I'd like to, ask Rick maybe to start on this. Are you certain that in your lifetime you'll be able to, to have something that's palpable, where you know how difficult it is to work with modelers because there's never enough truly molecular but there's never enough truly molecular data for them, and they always tell you, oh, I would have worked with you, but go fetch and then come back and we'll talk. And truly to be interpretable and to really go without the whole animal, we do need those levels of, in silico models, either very molecular or higher scale, on a tissue level. And do you think that there ever will be enough data or computational power or brain power to grasp that complexity?
Let's suppress this. I'm hopeful that, say, on the 5-year time scale, we will have a coarse model for mucus clearance based on an understanding of rheology and propulsion mechanisms over the long. I think that there will be issues, though, that will continue to be really challenging in terms of the polymer physics of the interfaces. I think we're closer to understanding some of the hydrodynamic issues. I think that the interface issues having to do with like adhesion of mucus to the surface, even in a physical context, are very challenging. So I think that might take another, few years to figure that out. In other words, how to really disadhese a mucus layer from the cell surface. To remain cautious, our biology colleagues who are grabbing a hold to another part of the elephant will continue to point out that there is still great specificity in a lot of those interactions, that mucins are not just kind of inert polymers on the surface, but they may actually play an innate defense mechanism themselves, possibly also participating in biochemical cascades with the epithelia. So there will continue to be biology in the sense of kind of binding and cell-based processes having to do with release that will, I think, continue to be discovered within that system. So I think that what I'm saying is I think many of the physical mechanisms we will continue to work out over the next 5 to 10 years. I think what will continue to happen is that there will still be biochemical specificity binding that's going on in that system that'll continue to be challenging. It comes back to a question I was going to ask the same question, and that is, what an engineer will always ask, it's part of an engineering training, is they'll say, okay, here's my task, what's the specification for success? So if our task is to come up with a predictive computational model that will satisfy the FDA or the EPA and do away with animal testing, what is the specification we have to reach? I'm not sure, I don't know the answer to that, not having been in those fields, but can anybody in those fields actually imagine writing a specification that a computational program of, who knows what it is, right? In the end, it's a black box. You feed in a compound, it spits out an answer, good or bad. What would be the validation of that box that would allow you to say, no, we don't have to go into an animal for this compound? The box said it's not going to be a problem. What's the specification? So I'd be delighted to hear if there's any comments on what would give people, in a validation sense, confidence that a computational framework was good enough.
Another good question. Let me also open it up to the committee and the whole audience, really, if you want to start weighing in on these things or ask questions. So one of the things about validation that I think is is important to understand is that we talk about validation as being a single spec, but really it's going to be context specific. So what Richard was talking about in terms of application as a priority setting tool is way different than a specification for full replacement. And so there's going to be multiple specifications, and given how Tosca operates now, there is precious little that's considered in terms of biological activity information for a lot of the chemicals. And so I think that this would be a huge advance already. And so the specification, if it were simply better than what we have now, might already be met.
So I'm going to continue to play devil's advocate here. So we're supposed to be talking about how this stuff will be applied, okay, which implies risk assessment. But what we're really talking about here is hazard identification or hazard screening. And there's a big difference. And so the point was, what is the end point? And I guess that was my question. What is the end point? I mean, if we're only screening in compounds for further testing, and that matrix is small, as you've said, what have we really gained?
I guess okay, so, the EPA has this backlog of 7,500, 10,000 minus 2,500 compounds that we're exposed to that have never had cancer testing or repro testing or whatever. Okay, so we can continue doing business as usual, which is nothing, right? Or we could say, let's make a stab at coming up with mechanistic predictions to say which ones of those look like they're hitting pathways that we know about that could lead to cancer or whatever, and queue those up and actually look hard at them. Okay, so that's really what we've gained, is we've gone from a situation where we don't really know what to do. How do you go into the next set of HPVs and MPVs that that there is no mandate to test, and so maybe people are being harmed, maybe they're not, we don't know. But to actually say, given today's best science and not waiting 20 years, so that's at the very least that's what we've gained.
But if you're basing regulatory decisions on that type of data, what if you use the wrong cell line?
No, I didn't say
you're not, at least prioritizing.
That's all we're doing, right?
Okay.
Go ahead, Chris.
There were a lot of things said that I really want to comment on, but I'll narrow it down to just 3 things. pretty quickly. Um, First of all, Richard, I'm going to disagree with your prediction. Entirely disagree with your prediction. I cannot possibly see that matrix as being sparse. That matrix is going to be completely crowded with responses across the board. Everything we've seen with the toxicogenomics database, everything we've seen with genetic alterations of pathways linked to human disease, everything we've seen with toxicology in the last 35 years suggests to us that the problem is going to be there's too much hitting on these pathways. There are going to be so many things hit. Finding the signal for the noise is what's going to be the key critical problem to looking at this. I'm afraid that assuming in advance that it has to be a big hit, you're going to miss some very important things. Assuming that you have to hit the beginning of a signal transduction pathway in order to see a change on the signal transduction pathway, I believe is naive. That pathway can be hit from number of angles. You can change protein function in the pathway well down the line from the signal, and you change the pathway response. So I think we have to remain entirely open-minded as to what we're going to do here. I think that's critical. There are good regulatory examples of use of this type of information. I don't know if Alex knows it, but EPA, FDA, NTP all use chemoinformatics as a first shot to set priorities for testing. Of course we use it. It's there. It's easy. There are a number of programs that are commercially available. And sure, we look at those. They tell us you might want to think about this, you might want to think about that. It's not the only decision. Other things weigh into that decision, but it's part of that decision process. When we think about these things that we're talking about here in the context of a decision process, not a decision, then it takes on a different flavor. You have to look at this. When you start talking about these things, we need to be talking about them, not that this assay or the 30 assays that they're doing in ToxCast or the 300 assays we've done in the high throughput screening at NCGC or the definitive answer. They're not. No competent biologist is going to base it on that. I think we have to think of it in a different context. The context I propose that we think about is that these tools tell us about things that act in the same way. We'll figure out potency afterwards. Let's find the things that act in the same way. Let's see what toxic things they act like. This is very simple. And then look at the ones that act like toxic things and see if they look like they're going to give us toxicity. That's a simple approach to the concept. That'll get us through to 10,000 very rapidly. And we can at least make a first cut that's going to be biologically supportable, and it's going to use as much data as we can. And then we can start talking about the more complicated stuff later on. And I agree with Ben, we really have to bring genetics into this, not only human genetics, but the animal genetics, because I think it plays a key role to unraveling what we see, and it's easy fruit for the picking.
Even though I'm supposed to moderate and be moderate, I think one of the points that we have to get to sooner rather than later, Chris, is this thing about let's leave potency for later. Because I think that the real difference between your view of the elephant and Richard's view of the elephant is that Richard was including potency now. And I think that the answer to the bet as to whether we're going to get thousands of hits or a sparse set is really going to be based on potency and whether these are achievable concentrations in a realistic physiological setting.
Just to be clear, just to make sure we're absolutely clear of what I'm saying we predict potency later. I would certainly, absolutely use dose response and potency in the molecular and toxicological range as part of my evaluation for setting priorities. But whether I would then use this information to go on and predict potency without doing any additional testing, I think is a second question that needs to be asked. I want the first question answered.
Go ahead, Linda.
I have a question for this group. I'm not really a toxicologist. When I started working with Steve Tannenbaum, I thought there was one P450, and was astounded to learn there's like 400. So this is a kind of open question. I was really struck by our project, stimulated by the Amgen problem of the comorbidity between inflammation and exposure to a chemical, a drug. And certainly we see chronic inflammation being linked very concretely to carcinogenesis. And I am not familiar with a lot of the models that are used, and whether you have any models that stress the system with respect to inflammation, because a person who somehow got, acute or chronic inflammatory process going on could be way more susceptible to some of these things than a healthy person. And I bring this up in part because my other life, actually the major part of my life, is regenerative medicine, and there's a lot of argument discussion in the field about the kind of models we use to test our tissue engineering scaffolds, because you test them in young healthy dogs, but you're going to put them in old people who've been irradiated and et cetera, or smoke or have diabetes, et cetera. So a general question is, and I haven't heard it brought up, is the interplay with the immune system, particularly because you can create enormous stress on otherwise perfectly happy cells by putting them in an inflammatory environment. And I haven't heard that really brought up, and wonder what everybody else thinks.
So one of the cell systems we use are actually they're human primary cells, which are in some cases put in a chemical context that looks like they're in an inflammatory situation. And certainly you get different cellular responses than when you don't have that. Okay, beyond saying, you can do the experiment, people have done the experiment. And it makes a difference.
It's routinely done as part of the assessment.
No, it's not
Okay.
routinely done. It is doable.
But the matrix might become way less sparse if you did that. Yeah.
Possible. Is your question on this string, or because I think Lauren's probably is. So,
Yeah, mine is just the metabolism component of it, and I'm just wondering in terms of even using them for priority setting without a better handle on metabolism. I really can't get as enthused as Chris can about them. So I just was wondering for a comment on being able to address the metabolism issues in these in vitro tests.
I mean, that's a huge, difficult problem. I was just going to weigh in with what Linda was saying, that 30% of the population is obese, and there's a strong correlation with inflammation and obesity. And so I'm very concerned that if you take very simple models, you'll never see the kind of comorbidity that a toxicant could provide in a human population. And so it's even a bigger confounder than metabolism, but I think that's a serious concern.
Could I make a comment that's related to the question of inflammation? I mean, we do have to think about what we're doing now, and I'm just not aware of any current, regulatory testing models. We use young, healthy animals when we test, and we know that there are people, animals that are more sensitive, and people may be more sensitive. And what we generally do in the regulatory context is put safety factors and uncertainty factors for that. We don't, but that doesn't mean that we might not be missing something totally. I mean, but that's where we are now. So we have that same probably have that problem more with what we're currently doing now than we do. We have the potential to.
They're probably extremely nonlinear.
So one of the things that, please.
I just want to comment related to that. With regard to mucociliary clearance, it's not clear if there's a good animal model for mucociliary clearance. I mean, you take a mouse model for CFTR, and it clears fine, which is we understand why in many ways, but it's obviously not a good mucociliary clearance model. So it might be, getting back to Ivan's question, a challenge for us to, and maybe in the sense of Deborah, we should say that mucociliary clearance is simpler than behavior. That in an integrative fashion, we could take mucociliary clearance and use it to understand how different biochemical pathways, which otherwise might be considered separate, and individual small hits would be looked at, that we could use this model to understand how smaller but simultaneous hits on biochemical networks would combine to create a fragile mucociliary clearance and maybe help populate the sparse matrix with compounds that should be looked at more closely.
So one of the just to put the optimist's view on things, which I am, is, you know, as Tina said, we have these models that are basically, you know, very genetically controlled, young, healthy kinds of models. But we do that because if you go beyond that without any information as to what your model ought to be different from that, you know, basically the number of combinations, the number of models that you have to test becomes outrageously impossible to fulfill. So what you need to do then, and what I think that what we're getting the data set that allows us to do is informed selection of models rather than one size fits all. And so to me, that's actually one of the potential strengths of a computational approach, a systems approach, is this idea of if you can identify what the likely disease outcomes are, then you can identify what the right models are.
To that, two
Sure.
responses to that. First, there was an NAS committee that actually weighed in on cumulative risk for phthalates and cumulative risk more generally, and took the approach that if we focused on the disease outcome and we knew what chemicals ultimately had been associated with that, we could work from the disease backward, and that in fact would define the mixtures that were of interest instead of trying to start with 10,000 and wondering which ones we ought to put together and why.
Mm-hmm.
So I think there's other ways of thinking about that, not that CompTox wouldn't contribute to it, because it can from a mechanistic point of view, but there are lots of ways to produce common physiological effects by very different mechanisms. And again, it goes back to this point that we do these studies in healthy male young organisms, and I think, you know, a lot of why we haven't been so successful in treating human diseases is because those models don't look anything like human disease. And I think we can all go out and look at the human disease and pick out some of the other risk factors we ought to be putting together with chemical exposures to have better models.
Let me get Alex and then Linda.
I sort of increasingly have a sense that we're mixing together two issues that should be discussed concurrently, but maybe separately. One is the issue of data. Second is the issue of data models. People like me don't have typically what's called in computational science domain expertise, such as toxicology domain expertise. So we analyze data and build data models, and we build the most robust data models we could. And we have our criteria that tell us that the models are robust. What we don't have is the utility of the models for serious questions that people who produce data ask. And I think there were a lot of illustrations of that. My specific point is that, yes, I could build very accurate model that will predict an in vitro assay, except the assay itself doesn't correlate with any in vivo endpoint. And so what am I working on in this case? We're using criteria that as statisticians we trust, but we have limited understanding of the significance of our model in the context of human disease outcomes. And so I think separating these issues and focusing on, I don't know, maybe it's correct to start from the disease and go all the way back and then talk to statisticians. Maybe we should be talking about it at the same time. My hope is that through this conversation, what will emerge is an iterative approach. People who produce data typically don't ask those who will later analyze this data how the experiments should be set up. So, you know, there is an opportunity for ToxCast to ask us about the compound nomination, for instance, for the next stage, based on considerations of chemical diversity, for instance. So I think it's the matter of maintaining this dialogue, but separating very clearly people who model data cannot provide answers to questions that the data doesn't address.
Thanks. Go ahead, Linda.
So I'm going to show that I'm an engineer and not a physicist. or a computational person in that a lot of what you do in engineering is try to make as few measurements as possible. So you often will have a system at an extreme state in order to understand about the system. And very common examples of that in materials testing, for example, we have advanced aging. You cycle it, you heat it up. In pharmaceutical formulations, you do sort of rapid aging by heating it up, et cetera, because you can drive the system into an extreme state and from that learn how the whole thing will operate. And all I'm suggesting is maybe, again, we think about ways to eliminate benign things by driving us into our extreme state, because every single one of us will be in some kind of extreme state at some point, probably in the next year. You'll have a cold, you'll get the swine flu, whatever. You're going to be in an extreme state. And are you more susceptible in that state to toxicants? So my suggestion is we think about incorporating the equivalent of advanced aging cycles of whatever that we do in all other kinds of engineering into the kinds of assays we think about here, because you can reveal a lot about the system by taking it to its extreme and then backing off.
Okay. You've been really patient.
Stephen McFadden. I'm author of a peer-reviewed paper on genetic polymorphisms in xenobiotic metabolism and adverse environmental response. I note that there are a lot of polymorphisms in the xenobiotic detox pathways, cytochrome P450 type 2D6 at maybe 7% of the population, and CYP3A4 at maybe 2 or 3% of the population, and so forth. My view is that these are not errors, that these exist because they offer species survival benefits. For instance, a lot of these are impaired detox pathways, but they also may activate carcinogens. So if your entire population is exposed to a high level of carcinogen, maybe these few percent will survive. So my view is that since you want your assays repeatable and your zebrafish are all genetically identical, considering genetic polymorphisms in xenobiotic metabolism and the variation in the human population must be done at a higher level. In looking at, for instance, Gilbert syndrome, which is impaired detoxification, on a table of where it was in the world, it was about 2% in South Sea Islands and about 20-30% in Africa. Well, glucuronidation requires glucose, but that competes with the glucose to run your brain. And in times of absolute starvation, if you have to walk 500 miles to get your next meal at the next river, maybe having no detox pathway isn't a disadvantage if you're in absolute starvation. So there may be species survivability benefits. There are reasons why these subpopulations exist. And these have to be reflected in your toxicological analysis at a higher level, because they're generally not reflected in the individual assays.
Thank you. Go ahead. Well,
Nobody's going to respond?
I think it's
I have a few points. First of all, I believe that metabolism has to be addressed. That's probably the markers that can translate the most between in vitro and in vivo. Without that, you can't compare them very well. Another point I would like to make, I need to point it up. I'm too tall. Okay. This is just right. Okay. Okay. Sorry about that. The other point is mixtures. A lot of things are not toxic by themselves. One of these things, melamine over there, that George knows quite a bit about. You have to deal with, there's a lot of things out there that we need to think about. The biggest cause of fatal medical errors out there is combination with alcohol or street drugs. It goes up a factor of 3,000%. Maybe we should start testing things in alcohol to see how toxic things are. I mean, these are one of those other things. Those are the two major points I want to make. And the last one I wanted to say is metabolism. She's correct. We can start seeing things in the dopamine pathway or the serotonin pathway when it's definitely not hitting it by itself. But we can start seeing things occurring in lower levels of tyrosine or lower levels of tryptophan and stuff like that. So just hitting just the serotonin receptor or the dopamine receptor is not the only way you can affect that system.
Go ahead, Ivan.
So I'd like to second on that and maybe move us to the next question here, which was brought up already by Chris this morning, which is where's the investment needed? Because as we hear all of these wonderful discussions, clearly there is a lot more to be done, and whether or not particular high throughput, high content, or even tissue, scale or computational modeling approaches can help us there or not, there's still probably not going to be enough resources and time and robotic hands, if not graduate students and postdocs, to fulfill all of those needs. And I am really interested in hearing from the panel where, they kind of abstracting from their own area of expertise, where they see the need as being the most acute. Because again, realizing that resources are scarce, are not getting any better in the foreseeable future, and us needing to move this field and the rest of the toxicology and neurotoxicology forward, where is the investment needed, being able to address all the most comprehensive needs rather than some of the more narrow-minded needs.
Why don't we start with a statistical point of view? I'd go to my statistician for this question. So Russ, when you were presenting your model, I mean, basically you had the Netflix, parameters across the top chemicals. Is there a certain size experiment that we're going to need to do before you can start doing the multiple regression analysis that allows us to determine which of those things across the top, parameters across the top, are really predicting?
I don't. It's really, we can pretty much adapt to whatever size data sets you have. I mean, the more data you have, the better you can usually do as a general rule. But I think the statistician, these are really tox questions that we've got tools that'll help you answer the questions once the questions are formed. To ask, it's typical with I refer to the power and sample size question. A lot of that depends on how much is a false positive worth versus a false negative, and that's more of a scientific question. That's not a statistical one. So it comes down to maybe developing loss functions or utility functions or assessment of. It's almost like risk assessment on this exercise.
Mm-hmm.
So Russ, you're saying all the statistical tools are there, no more investment, right? So just bring more data.
That's what I heard.
They're ready to run.
So you're gonna have to get your salary by winning the next Netflix prize. Go ahead, Alex.
I think I brought this up before, but I think the real challenge is creating a framework where computations iterate with experiments. Because I don't think there is a workflow that really allows this to happen. We explore data sets, we publish papers, there are hundreds of methodologies, we sort of compete or compare methodologies applied to the same data set. There is no moving forward. It has to allow in predictions to guide the experiment and allow it and I thought point that Rick brought up earlier, which is what the specs are, is of extraordinary importance. Because, we need to know how much we're allowed to fail in making predictions and what the level of tolerance is with the level of predictions. High throughput screening methods are used routinely and the hit rate is 0.01%, and that's acceptable. If I publish a prediction that will have a 1% accuracy, that actually will be very bad. I couldn't publish. So, what the real criteria are as far as using models in a prospective way and sort of learning from each other. I think that's the most important aspect. And another point I want to make is again, the data analysis is data driven. So back to the matrices, are they sparse or not? The matrices that I analyze are definitely sparse. PubChem has 300,000 chemicals that have been tested in the last 5 years. 54 compounds have been nominated as probes by NIH. That's the sparsity of the matrix as far as data dictate. So again, the definition of a probe comes from experimental scientists. So our models should agree with experimental restrictions, and that's something that we need to know. and that does require investment.
I'm not going to turn down money. So what we need to develop good enough models. In some ways, we need to fill in between the pursuit of basic science-based models, principled models, and models which are making a bunch of guesses in trying to get to something that's better than what we have now. That's a very flexible way of thinking that we don't do a good job of training. And so I think we need to, that's why I constantly put training up on these slides about what do we need to do. So I think we need to take some of the basic science modelers and the engineers and have them trained so that they appreciate the fundamental science that continues to need to be done, but appreciate the imperative of having better tools for decision making. And I think the decision making side of the science, whether it's the clinician or the regulatory agency, is now too divorced in some sense. I'm going to get in trouble here, I sense, from the basic science modelers. And so that's why I had it up on one slide. I skipped it. We need to teach the imperative to some of the modeling that goes on in academic settings so that they can understand how what they know can create better tools now, sooner.
I agree with you. I think more and more we see the cadre. You mentioned in your talk that it's hard to get, you have people who are good at the math and good at the biology, but I think more and more we see programs across the country trying to blend those two. We've actually I led development of MIT's first new major in 40 years. It's in biological engineering, where we start from the ground up and really teach students how to do engineering based on molecular life science. It's going to take a while for these people to filter up to the kind of decision-making positions where they understand the biology and also a lot of the analysis, but I think it's starting to happen, and unfortunately NIH doesn't foster it as much as NSF. NSF has been a real leader in trying to foster this, less so NIH on the training side.
Go ahead, Abby.
So when I listen to Chris, you're saying that you're in hazard identification and that you want to try to figure out what you can do with the database you've got. And I look at Toxcast and some of the approaches that have been done. I don't think we need better statistics on bad data. I think we need better data from which we can maybe apply statistics. And I'm very concerned that we have to think ahead of time. If our ultimate goal is regulatory, then what do we need to start populating? I noticed in the Toxcast, they tried to, I think you did like 1 for doses less than this, 2 for a dose. I mean, you had some criteria, and can they match with the other folks that are entering it on the other side so that eventually they all can merge? I don't feel like it's connected. And the criteria for entering data in is going to be critical if Chris is going to have any luck with finding any correlation statistically.
Right.
So that's where I think the money should be. And you've got curators who are students who don't know anything about really weight of evidence toxicology or anything like that. So you lose all of the information that's really there.
I'll let you defend Toxcast.
I'm going to rebut. I absolutely agree. If the data, whether it's the in vitro data or the in vivo data, is garbage, it shouldn't go in the database. Okay, and in fact, for everybody who's been looking at the Toxcast data and wondering why it's taken us so long to release it, it's because we've spent a lot of time trying to understand what good data and bad data is. Okay, on the in vivo side, where we've had all the student contractors working, they've worked under supervision of old-time toxicologists. The data is what the data is. We wish that the data, all the in vivo data was perfect, but it's not. It is what it is. And I think what's in the in vivo database, the ToxRef, is as good as it's going to get.
Yeah, well, and the devil's in the detail, because what you probably are starting out is with the DERs, right?
Yeah.
And those are the standard studies. And the things where you have all the interesting data are the non-standard studies, from which I'm not sure you always have DERs. So the richest, best data that we submit to regulations is that has actually some maybe mechanistic chasing after may not get into your database.
And that's not a science constraint. That's a political constraint.
Well, but I'm just saying that we have to bring people like us together to examine what you're actually putting in and say, are we really missing some of the most richest, most important data that will be useful for your data mining?
What's an example of that for those of us not
Well,
in the,
you know, like you might chase
don't know
a
what
kidney
DER
with
is.
an alpha-2 globulin, mechanism, or you might chase something else with some other kind of mechanism, and you would submit a non-guideline mechanistic study that can range, it's just a wide range of different sort of things that go in. They're the most interesting, important information.
I got a long line of people. Marty?
Marty Stevens with the Humane Society of the United States. I come at these issues primarily from the perspective of the NRC report on toxicity testing in the 21st century. And I know that's not the primary focus of our workshop, but it was mentioned several times this morning as a jumping off point. And I think that report is far more revolutionary than one would guess from hearing some of the presentations. Now, just a couple of points related to that. The report, to me, focuses very much on human health and human data. It's not for priority setting and animals. Granted, there are few human data to choose from, but I think I like the notion I heard this morning of sampling different parts of biological space, maybe not only molecules and cells, but tissue slices, in order to whatever one can do relevant to people that would confirm your body of evidence that something is indeed going to be a problem. The report is essentially a recipe, albeit a general one, for a very different way of doing risk assessment. So it's not a hazard identification report, and I think we need to figure out ways to integrate this new kind of data into a meaningful package where you say, even though I don't have all endpoints modeled, I don't have the DNT and other things modeled, I have enough early response data to indicate that this chemical is going to be a bad actor. And the third and related point is that the NRC report talks about not only the primary testing, the kind of thing that maybe Chris Austin talked about, but also targeted testing, sort of secondary testing that one would think to do after looking at the results of primary testing. And it's there that maybe we can use some of the more apical methods that were mentioned today, zebrafish and other things, as follow-up to what the molecules and cells bring us. So there's room for an integrated testing approach, even though the report focused heavily on lower levels of organization. Thank you.
Go ahead.
I wanted to try to answer Yvonne's question with how should we spend money, and I think we should take a two-prong approach. One is to understand the limitations of these simpler systems, for example, C. elegans or zebrafish, but also I think we need to understand a lot of the cell systems that we're already working in. We know that primary hepatocytes give you more information than HepG2 cells, but we learned from Linda that you got to put those hepatocytes in the right milieu. Add fibroblasts, they behave differently. And I think some of the computational modeling could come at that problem and say we know that hepatocytes primary have these good aspects, but they don't recapitulate the liver because of these. And so I think knowing the limitations of the system would allow us to understand the quality of the data. I don't think the data would necessarily be good or bad. I think if you don't use the right system to ask the question, it just doesn't get you to the answer. So I think we could put money into both of those fields by exploring the limitations of the system, understanding that you don't want to ask sophisticated neurological responses from yeast, but yeast are good for this, this, and this. And you could screen a whole bunch of compounds knowing that's the space you're working in.
Good, Doug.
Doug Wolf, US EPA. I guess I follow up my Humane Society colleague, although it wasn't planned. I think we need to define what the goal is. So if the goal is really to fulfill the lofty ideas of the tox testing in 21st century of being able to do quantitative risk assessment based on a set of in vitro assays and in silico models, then how you answer these questions will be very different than if your goal is to be able to look at a large set of environmental stressors and say, we need to understand how these might cause human disease and under what circumstances might they cause human disease. And so that's a very different approach and a different answer to those questions than saying we want to get to where we don't use any in vitro, any in vivo assays, and then say, well, under what circumstances and what kind of targeted testing do we need to do both in vitro and in vivo to answer the conditions under which we would anticipate human disease. And then there will be some endpoints which in vitro models just won't be satisfactory because of the nature of the physiologic system you have to address. And you'll have those types of in vivo studies specifically to answer those questions. So I see those as two very different goals. And I would like to know what the panel is thinking of, well, which goal are we going to head toward? And then once you have that, then you answer the questions in that framework, because the investment for one is going to be very different than the investment for the other.
Reactions?
Well, I think we have to remember a lot of this is EPA driven. They are a regulatory agency. So ultimately they have to do the risk assessment. So I don't know. I mean, it's really theirs to answer, but if that's the goal, then risk assessment is about protecting public health. So
I agree with Doug that one has to keep these questions separated in mind. However, that doesn't mean that the approaches that we were discussing today or what was recommended in the Toxicity in the 21st Century report are not necessarily should be different. I think that the interpretation of the data might be different for different applications. However, I agree with Deborah. I mean, it's the data will be produced and the models will be built, and then the regulatory agencies will have to decide how to use them. Is it one chemical at a time? IRIS document, 600 pages of pathway-based risk assessment. Here you go. Or which of the 10,000 chemicals that are high production, medium production volume, we really have to worry about. So again, I agree with Deborah. This is, the regulatory agencies have to decide, but we're not going to come up with new methods. The methods are what they are. The models are what they are. It's just how you use them.
Bob.
So Bob Kavlak from EPA. So my 2 cents, what do we need to make this work? I'm going to agree with Chris, so this is a rare event. It's getting these bioactivity profiles and see if we can match them up to known toxicants, and then look at unknown chemicals to see if we can find their activity profiles and then make the prediction. Because if we can't crack that nut, which is a pretty low bar actually, I think, we're not going to get these assays into quantitative risk assessment. When we started ToxCast, we just took off-the-shelf assays. We had 5 years the agency gave us to try to make a difference. And so we weren't going to get into, let's develop methods, because we'll be sitting here today developing methods. And so what we did is we went to pharma and we kind of said, what is available for us to buy off the shelf now? So those are the assays we got. I mean, we kind of went as broad space as we could, but it was still, this is what we could buy 3 years ago. If I had to say what's the most important thing to do right now is I'd go to the domain experts, the neurotoxicologists, the developmental neurotoxicologists, the reproductive toxicologists, and I said, tell me your 10 most important pathways and tell me the cell context that we need to screen those in. And maybe you're going to come up with 20 assays, 20 pathways. I don't know, but there has to be some priority setting by the domain experts that these are the critical pathways that when chemicals perturb cause effects in my system, whether it's inflammation in a hepatocyte or dopamine cell survival in the, some nucleus. That's what we need. If we get that, we can develop the assays that are going to make this better. But it's really getting better data to give to these kind of guys and Russ who can do better models with them. But, 5 years from now, I think we'll be laughing about what we're doing now. But we need those pathways and we need assays to do it. So my 2 cents.
Thank you. Yvonne?
Just to respond to that, I do not disagree that trying to identify major classes and then try to bin unknown things into known classes is a bad idea. This is what toxicology has been doing for probably more than 200 years. However, I do worry a little bit about where the high throughput screening is going with this, because this clearly hasn't been very successful, but was the promise of toxicogenomics, the whole compendium approach. If we can just, give these chemicals and then build a database large enough, then we give an unknown drug and then try to potentially see what the liabilities are. That hasn't been very successful, so I'm not sure that that should be the sole goal, or we should benchmark us against that particular application.
I take issue with the fact that it hasn't been that successful. I mean, I think it's in widespread use in terms of looking for some sort of toxicity profile or some sort of gene expression profile that predicts toxicity. So I think it's a matter of how you weigh success. Yeah. Go ahead, Chris.
Abby, you got me thinking. That's always a dangerous concept. What would I do?
Good, that was what they told me to do.
What would I do today? Today, you give me the money that I was thinking about in 2003 when we started looking at this for the NTP and thinking about where we would go with it. What would I do today to address the question? Well, part of it is what we just published, and I would suggest you look at the paper that Carolyn mentioned, where Julia Golke was the first author, because there we asked ourselves a very simple question up front. Causality. We want causality in humans. How are we going to get that? And we thought, okay, if we're talking genes and pathways, the best place to look at that is polymorphisms in humans related to human disease. What genes are those polymorphic genes that relate to disease? What pathways are they associated with? Suppose we have chemicals that mess with those same pathways. Okay, that seemed like a logical first shot for us. And we learned a lot from that exercise, and I think that exercise is still a viable exercise. Not just with human data, but with the rodent data. There's things you could do with that would open our eyes to a lot of understanding between pathways and when we perturb them, what they really mean to disease in a causal way. So I would invest in that area, and NTP is. We're doing the haplotype mapping of 27 mouse strains. We're going back and thinking about doing a full genetic mapping for a lot of different possibilities in those strains. So we are investing in that, and I would still invest in that. I think that's important. Technology changed. We couldn't do toxicogenomics on fixed tissues, formalin-fixed tissues. We can now. Well, whoa, wait a minute. If you had this for me in 2003, I probably wouldn't have done high-throughput screening. I'd have invested my money in taking every single 90-day study that the NTP had done, which are about 500 of them, for which we have formalin-fixed tissues, and I asked myself, can I use a genomic signature from these tissues to predict what I saw in the NTP's 2-year chronic exposure bioassay, or the NTP's developmental tox study, or the NTP's neurotox studies, which are all there and all available, and ask myself, can I see signatures here? That might have been a better investment for us at the time, and it may be a better investment now. Today, metabolomics has changed the face of what we're looking at in terms of metabolism, not just of the chemical itself, but changes in cellular biochemistry. I'd probably invest in some metabolomics at this point, not necessarily the high throughput screening. So the window's going to change as we go along, and we have to be, one of the big problems as a statistician now, instead of as a biologist, one of the big problems I see is we never get a full database. We get pieces and parts. One of the most annoying EPA science advisory boards I ever sat in had to do with PPAR alpha, because it was the argument was being made that it was not a human carcinogen because of all this stuff with liver and PPAR and liver, etc. But it was piecemeal, a little bit here, a little bit there. There was some contrary findings in monkey that were thrown out and ignored. There were all sorts of stuff there that there was no balance to the database. So one of the biggest problems is if we change path now and don't continue on trying to do some of the things we're already doing, we're going to cut that database short and all of a sudden start on another one. So those are difficult questions that have to be addressed and discussed. But you did ask me where I thought I would go now, and I would have changed things today as compared to where we were looking 7, 8 years ago.
Something.
Sure.
I think that there's a nice segue to increasing communication between a lot of these very large efforts, because the technology is going to continue to change and the data is going to continue to change. And I agree that funding is a really tough thing, especially if you're not a government agency, to keep these resources going. And I think if there was some other mechanism to keep these resources coordinated in such a way, then we can keep these things integrated, we can avoid redundancy, we can continually look at ways to better integrate these data and look at how they might be able to complement one another. And rather than all of us sort of, working like this, then I think we would do a better job to see where the holes are, where we can fill the gaps, what questions are now being asked that weren't being asked 5 years ago, and do we have data we can leverage to answer that in a different way, or do we need to invest in different assays? And I think there's a real disconnect between some of these resources. And just as a small example, I like Ben's comment about integrating data from like ZFIN and C. elegans, and I've been trying to do this for many years with some of these databases with CTD, and Monty Westerfield is a good friend. I like him a lot, but I contact him almost every year and say, let's integrate ZFIN and CTD, and he talks to his group, and they come back and they're like, we really don't understand how toxicology would fit into ZFIN. And so, I think there needs to be some formula or some mechanism for getting groups together, because I think the more inclusive we are, then we're gonna think more outside the box about what data actually can help us answer some of these questions.